here today to talk about genetics and epilepsy. I'm Nikki Baker. I'm with the foundation, the Epilepsy Foundation of Minnesota. I've been here for 20 years and just worked in various capacities. And next up is Dr. Smith. We are so thrilled to have Dr. Smith here today. Like I cannot tell you how much I loved working with him. And Dr. Smith is a pediatric epileptologist at the Minnesota Epilepsy Group in Roseville. He has earned his medical degree from the University of Medicine and Dentistry from New Jersey in Newark, New Jersey. Dr. Smith completed a residency in neurology at the University of Pennsylvania in Philadelphia and a residency in pediatric neurology and a fellowship in a pediatric at the Minnesota Epilepsy Group in 2016, which I want you all to know was that most states don't have any pediatric epileptologists anywhere near them, so we're extremely lucky. Dr. Smith founded the Epilepsy Diagnostic Clinic within the Minnesota Epilepsy Group in 2016 and continues to serve as a director. He currently is also the director of the Tubercular Sclerosis Center of Excellence based in the Minneapolis, the Minnesota Epilepsy Group, which, as you know, there's a lot of co-occurrence with TS with epilepsy. Dr. Smith is a clinical researcher with experience working on several clinic trials and writing various medical papers and journals such as Neurology and the Journal of Child Neurology. I cannot tell you how thrilled we are to have him here today. He is, as far as I know, he knows genetics and epilepsy like a glove, and so we're thrilled to have him here.
All right, put the pressure on me, Nikki.
Yeah.
We've been doing this together for many years now, and you keep inviting me back, and that is what I'm taking as proof that hopefully I'm getting what people are interested in hearing about. It's always important. I always start off with this slide, and I don't want to just gloss over it like many people do. It's an important thing that I want to make sure is clear before we get started. I'm going to be talking about genetics quite a bit. I'm going to be talking about ordering genetic tests, and before you listen to anyone in medicine, you always need to know where is their money coming from. I'm not getting a cent. I get nothing financially, either from the Epilepsy Foundation or for doing these genetic tests. And so it's clear that when we're talking about, it's not because that I'm making any money off of it. This is going to be because this is what I do for a living, and because it's really, really interesting stuff. And I hope that you find it as interesting as I do. I can advance. Only an hour on this is a very tough topic, and I see a lot of the same names on here that have probably been to some of my previous lectures, and so I hope it's not too elementary, but I want to make sure that I cover the basics, because a lot of times when I see issues, it's a lot of confusion about the basics, and everyone thinks they know genetics, but I don't think they always know exactly. And I want to make sure we're all on the same page. So we're going to be going fairly basic, and each one of these questions I could easily spend an hour on by itself. But my goal is to get you as excited as I am and give you just a little taste about what we're doing. The first is really going to be about ground setting. It's just first understanding, what is genetic testing? Are all genetic tests created equal? Is a genetic test today the same as a genetic test 5 years ago? Obviously, the answer is no. Where are kind of some of the blind spots on these genetic testing? And again, a little bit of a primer about some of the confusion that comes in with it. I'm trying to make this as useful as possible, which is always really the question for most people in this audience is going to be, is this something that we should add? Is this something that we should do? Would genetic testing serve any useful purpose for me or for my child or for my loved one with epilepsy? And I'm going to give as practical an answer to that as possible. I'm then going to kind of start talking about what we don't know. It used to be I had to convince people of the usefulness of this. Now it's the opposite. Now it seems like everyone wants it, everyone runs on into it, and everyone seems to think that our genetic testing is perfect. And so I want to start by actually highlighting what we don't know and the limitations of our knowledge before I start talking about, well, okay, what do we actually know? And giving some really concrete examples to kind of really drive the point home. And then I'm going to start talking about the little bit of the future. And I think that for many people, this is really what they're most interested in. And so it's a little pie in the sky and a little bit maybe perhaps overly ambitious or optimistic, but I think that that's what people are, especially again, for patients and or people who have loved ones with epilepsy, that's really what they want to hear the most about. So we can advance. I apologize. I have a lot of fun travel photos, so I love including them. So let's start off again. Let's start off with the question is, what is genetic testing fundamentally? Next slide. And that has to start off with answering what is DNA? And when I even say genetics, what does that even mean? Now, essentially, genetics refers to the building blocks of what makes you, you, and me. That every single one of our cells has a set of 46 chromosomes, more specifically 23 pairs of chromosomes that all contain all the building blocks of information for you and I. And you can visualize that information. This is something called a karyotype. This is actually one of the forms of genetic testing, one that's very rarely done anymore. And so this information is effectively like a whole bunch of books. So again, 46 books, 23 pairs of books. You can advance to the next slide. And those 46 books contain a massive amount of information. You can go to the next slide, Nikki. Contain a massive amount of information. If you kind of did this in standard print in terms of like each letter of the code that is in your DNA, then and you typed it in regular font print, it would be approximately 200 feet high. And so this is a gigantic amount of information that is encoded, again, in every single one of our cells. And this information then gets passed on to our children, or at least half of our information gets passed on along with our partner who winds up contributing their half. In the process of making a new individual, there is on average 6 typos or 6 changes or 6 mutations, is all meaning the same thing, 6 changes that wind up happening on average. While they're copying over, again, those 46 books, these typos wind up happening. And this is what is responsible for all of the changes that we have seen in people over time. And again, why all of us, they always say, has a unique genetic code. Next slide. And so I'm going to use a number of metaphors as I start talking about genetics and make sure, again, you'll hear a lot of people use some scientific terms. I want to make sure that when I use terms, you know exactly what I'm talking about. And I think that the book metaphor works very, very well. And so each chromosome, in this case, is one book. Your genome, which is, again, all of the information that makes you, you, is 23 pairs of it, a total of 46 of these books. Each gene, and by gene, I mean something that is actually read that tells the story and makes protein more specifically. Each one we're going to consider like a sentence. It's a series of letters. And the length of these is really much longer than a sentence. It's probably more accurately a paragraph. But we think of it as a sentence because each one is going to be a series of 3 letters. And each letter is going to be A, C, T, and G. Those are the only ones that we have in our DNA. There are other ones in our RNA. I don't want to get into that. Our DNA, again, the part that actually keeps all that information, is a series of ACs, Ts, and Gs in three letters in a row. So like CAT, GAT. Each one of those, think of them as individual words in that very, very long sentence / paragraph. You can go to the next page. Now, why does all this matter? Why am I spending all this time giving you the basics of what you had back in a high school biology class? Well, because a lot of times people will come to me and say, I already had genetic testing, but they don't really elaborate beyond that. And we have to go work through it together to figure out exactly what was done, because not all of it is created equal. As I had mentioned, karyotypes are the most basic elementary form of genetic testing that we can do. It just shows you what you can see, generally speaking, what your DNA looks like. It's a surprisingly labor-intensive process to do this. And it really only shows you huge changes in your DNA. We don't really read a single letter in those books. We have no idea what's inside of those books. We just know that there is either a missing or an extra book, or the book looks odd. So what are good examples of that? Well, Down syndrome is the most classic example, which we call trisomy 21, meaning that you have 3 copies instead of 2 of the 21st book. Less commonly, there's something called Edward syndrome, which is trisomy 18, 3 copies of book 18. And rarely, something that the only reason I get karyotypes these days is something called ring 20. It's a rare condition, and where you can have that one chromosome at the bottom, number 20, where the 2 ends stick together. And instead of getting a line, you get a circle, which causes this particularly hard to control epilepsy. Let's do the next slide. Now, this has an example of an abnormal karyotype. If you look down at the bottom, count all the way to number 21, you're going to see 3 little blobs there where there should ordinarily be 2. And that is where we have trisomy 21. This would be someone with Down syndrome. Again, I have no idea what any of the words are. Every single one of those books could be blank, so to speak. Obviously, they can't be. Otherwise, you wouldn't actually have a person to take this karyotype from. But it doesn't actually spell any of it out, and it's not going to pick any subtle differences between them. There are other things that scientists can do. You'll notice there's like some black and white and gray streaks to that. Scientists actually count them and have recognized patterns to it, where they can get a little bit more information. But fundamentally, that's all we really know about this, is whether or not you have all of your books. Let's go to the next slide. Yeah, the arrow on this one, if you look at number 20, number 20 there, that is an example of ring 20, as I had mentioned, this other rare epilepsy syndrome. Sorry, we can go on to the next slide. Now, this is a much more common one that many people, and probably people in our audience, have had or have considered having done. A chromosomal microarray kind of used to be the initial screen test for genetic testing, and it's one of the more common ones that used to be done because it's relatively cheap and easy for people to do. Another name for this is called a SNP array. I don't want to get into the acronyms, what they all stand for, but the entire point of this is that, again, we are not actually looking at the letters. What we're actually looking at is the total amount of DNA effectively in each one of these chromosomes. And so really what this does in a very, very crude manner, and this is not a perfect metaphor, is that it's count. And so if there are any geneticists in the audience, I know you're going to wince when I do this. But if you're a geneticist, I would think you're not coming to my lecture anyway. The closest metaphor to this one is that it counts the pages in the books, making sure that there's not a significant number of increased or decreased pages. So we know ordinarily that this book is about 4,000 pages long. It turns out actually small deletions don't matter a whole lot, that depending on where those missing pages are. But it's really counting and gives you a general idea as to where those missing pages are, in which book, in which chapter, so to speak. And so the next slide just kind of gives you a general idea as to what it is. They wind up essentially putting a reference genome on a chip, and then they compare the patients to it, and they see whether or not there is relatively more or less of the reference DNA. So I'll go on to the next one. Now, the next one that's easiest to understand is what I think everyone kind of thinks of when we think of genetic testing, but this isn't quite commonly done. At least we can do it, but it's certainly not a regular occurrence. There are special cases where we do this. And so can we just read everything? Can we read every single letter in every single book and just spell out that giant stack of information? And the answer is yes, we can do that. And that's called the whole genome. Now, whole genome, again, is done not very frequently. I have probably maybe 24 that have gone to this point of testing. More commonly, we'll do something called a whole exome. So whole genome, again, looks at every single letter at all of those pages with certain exceptions. The exome, by contrast, only looks at the parts of these books that is actually read. Now, again, imperfect metaphors, but it turns out that 99% of the letters, all the pages in those books, don't actually mean anything. Your body never actually reads them, never turns them into protein. And so the way that we think of it is this is probably a table of content, footnotes, references, things that don't necessarily tell the story, but do help you understand the story. And they certainly play a role in telling your body when to or which cells in your body to express which of these genes. But the majority of it is really not very well understood. And so we don't sequence that with great regularity. The biggest challenge with this right now is actually interpreting it, just because we don't have enough people that have done it where we know what normal variation looks like. As I mentioned, everyone in this room looks different. And so unless I have a twin brother I don't know about in this audience, no one looks like me. And science is not quite yet well equipped. We have tools to answer this, but we're not perfect yet at figuring out when a difference is an actual problem, as in causing epilepsy, or is this just a difference that makes you, you, and me, me? And so one of the big things we do to help us figure that out is that every time we do either whole genome or whole exome, we're also sequencing mom and dad. And the reason for that is that we are looking for those typos. We're looking for those very unusual mutations, those unusual changes that focus on those parts, because it's more likely a problem came from something that is new in the DNA. Let's move on to the next slide. Now, what most people get is not really any of these. But right now, what pretty much the vast majority of epilepsy is epilepsy panels. Now, when I first started the clinic 6 years ago, this was extremely expensive. It was about $5,000 to get this epilepsy panel. You would get about 150 genes, so it would take about 3 months for this to come back. These epilepsy panels are vast majority of the time what we wind up ordering these days. And epilepsy panels are basically, they're picking specific sentences. They're picking specific genes that we already know for other reasons. They focus just on the sentences that we know already cause epilepsy. Now, this is helpful for a number of reasons. It makes us understand better what these mutations can cause. It helps us understand whether or not there's a relationship between this gene and the seizures, and makes it much, much easier to interpret. It also is dramatically cheaper. And for people under the age of 8 with epilepsy, it's actually completely free. And this has actually been underwritten in a wonderful program, which again, I'm not going to advertise for anyone, but the point is that it's free. And so it has made my job so much easier, but has contributed to what, as I had mentioned, now I think everyone gets this. And now I'm dealing with the other side of, well, where are the blind spots? There are actual blind spots for our genetic testing. Anyway, the current iteration of this has about 300 genes in its regular panel, and it doesn't require blood. It's something that we often do in the office, just swab your cheek or you spit into a tube, and we mail it away and we get an answer back in a few weeks. And when it's that easy, as you can imagine, pretty much everyone wants it and pretty much everyone is getting it when it comes to epilepsy. If you're over the age of 8 and you're interested in this, the vast majority of time it is covered by insurance. And if it's not covered by insurance, typically the out-of-pocket cost is in the range of around $200, $300. And so it has gone, again, in 6 years from very expensive to this is a reasonable thing. And most people I talk to are very curious and are very interested in this information. Generally speaking, at this point, it's really just a standard epilepsy panel. And so we don't even need to go by subtype. It's really only broken down in a few specific cases where, let's just say that the child is both epilepsy and intellectually disabled or autism. There are other panels that look more at the intellectual disability or autism genes. But the point is that they're also very accessible and very easy to get done. Let's go to the next slide. And so this is where I had mentioned where we get often into a little bit of trouble because the answers aren't always clear. And sometimes no data is better than bad data. So let's get to the next slide because this is an important point, and probably I actually had multiple patients this week where this was a tremendous focus of our conversation. So even doctors get this wrong all the time. And one of the struggles with epilepsy or with genetics, I should say, as I just mentioned, is we don't know what abnormal always looks like. We're often struggling and saying this looks different. Our technology is well set up to say there's something different about this particular person's sequence background. It doesn't look like 99.99% of other people who've had their DNA sequenced. And so when that happens, right now, there are some algorithms that they use, effectively Google Translates, that run the change through their Google Translate, and they tell us, is this going to actually cause a problem? Is this going to cause the sentence to not make any sense? The metaphor I like to use for that one is it changing a CAT to a KAT, where you and I can just read right over that, and we already know exactly what that sentence means. It turns out your DNA has a lot of redundancy, has a lot of things where those changes can happen and it's not a big deal. And so we are struggling at recognizing the difference between a difference that is just, again, something that makes us unique versus something that is actually a problem. Next slide. And so, again, I'm going to keep highlighting that point because it's a simple point, but it's such an important one to understand. When we come back with answers, 9 times out of 10, what we're highlighting is it looks different. We're not necessarily saying this is causing a problem. And if it says a VUS on there, a variant of unknown significance, we're not actually saying it's a problem. We're saying we're not sure if this is a problem. And so this is where a lot of the question of should I get genetic testing comes in, which is do you have a team in place that is prepared to actually do the homework and figure that piece out? Or are they going to have someone's going to say, the test says this, it's got to be that. And I've seen some really smart doctors who often just make that assumption where they see that and they just say, okay, this is it. And so when they actually do the homework later on and, read the literally hundreds of pages that's on this gene, I mean, it takes a lot of work to get there, where they recognize, oh, wait a second, this doesn't quite actually make sense for my patient. And so I like to make it really simple because most of the stuff that works consistently is simple and easy to understand. The first thing is just asking, well, is this new? All right, do other family members have this? So if let's just say, if I had epilepsy and I sequence my DNA and I have a change in this one gene that causes epilepsy that looks just like mine, but my mom and my dad don't have epilepsy. Well, I already know that half of my genes came from my mom and half came from my dad. We assume, of course, I look like my dad, so it's likely that the paternal is for sure there. But we would test them to see, do they have that same mutation that I have, that same difference that I have? And if one of them does, and they don't have epilepsy, well, now that's a red flag to say, we now know one other person who has this same change and doesn't have epilepsy, meaning this change is now more likely to not be a problem. Now, on the flip side, let's just say that, again, I have this change in this one gene and my mom and my dad don't have it. Well, again, either I'm not testing the right dad, or this is a typo that happened when I was formed. And as I had mentioned, not a whole lot of these typos happen in the process of forming a new human. So boy, that would really be a heck of a coincidence to have a typo in this gene and to have epilepsy like that, unless it's a problem. And so it's a strong, strong proof that this mutation is causing my problem. And we have a special term for that. We call that de novo, which is just Latin for new. It's a new change. And it is probably one of the strongest things that we can say that this is likely the cause of your epilepsy. The next thing that we look at is, well, what does this gene look like? And I spent a lot of time on this, and this is really where I think a lot of the confusion happens. If I test someone who has myoclonic epilepsy, a certain type of epilepsy that has twitches to their seizures, and they come back with a gene mutation that's associated, a variant in this particular case, in a gene that causes frontal lobe epilepsy. Well, the patient doesn't have frontal lobe epilepsy because their genetics comes back with that. the patient has myoclonic epilepsy. And this is probably the number one problem that I see with genetics these days, is that a lot of doctors are letting the genetics tail wag the epilepsy dog. Now, what do I mean by that? When the tail wags the dog, it means we're getting the cause and the effect mix up, that there's a disconnect in between those two. And they're seeing, okay, this gene has this type of seizure associated with it, so you must have that. But the answer is you don't have to, actually. It actually could just be we don't understand that gene well enough, or this gene isn't the problem. And if the patient or you have a different kind of seizure, a different kind of epilepsy, that dog trumps that tail. And I don't care about what's coming on that genetic testing. I'm more focused on you and your seizure type than on that genetic change. And so we have to put those two of it together to actually make sure this makes sense. As I had mentioned, the next step is, what does the computer think? That's the Google Translate I had mentioned. And then lastly, just, does this make sense? Does a type of epilepsy sound at all like we have? And more importantly, do I need one bad copy of this gene, or do I need two bad copies? I'm still surprised at how often doctors make this mistake. Bottom line, that for most genes, for every gene you have two copies, with the exception of ones that unless you're a male and one's on your X chromosome, you only have one X chromosome, you only have one copy of those genes. Every other gene in your body, you have two copies. And for most genes, you need two bad copies in order to have a problem, because when that one goes down, the other good one can do all the work. Now, for many of our epilepsy genes, that's not true. You only need one bad copy. But if you see on there where you need two bad copies to have a problem, and you or your loved one has one bad copy, that one bad copy should not be causing a problem. And you can, for the most part, ignore that, with certain qualifying caveats to that one. So does it make sense? Does this variant actually need two bad copies? And if it needs two bad copies and you only have one variant, you don't need to worry about it. Let's move on to the next slide. And I promise there's going to be time at the end, so because I know I'm talking about so many things. So again, I just highlighted so much about what don't we know. And this should be like, wait, shouldn't I be like, hawkish on getting genetic testing on everyone? And for my patients, the answer is, for the most part, yeah, I'm a big fan of it. But I want to emphasize, before we talk about the benefits of it, let's just talk about what don't we know. Where are our blind spots? Next slide. There's a reason that we have these blind spots. And I think this is easy to understand only if we talk about the historical perspective as to where we're at. And I was in college when we finished the Human Genome Project, and I still remember that the pompous fanfare that we all thought that this was going to be like revolutionize medicine, revolutionize our understanding of the human body. But it took them 13 years to do that first human genome. And it took over 200 academic centers and 20 institutions, and it cost them $3.8 billion to do this, which, believe it or not, was significantly under budget. It was about double was how much they were expecting it to cost. So the very first one that we had ever done was only done 20 years ago. Next slide. Now, this was actually an article from 2019. The entire country of Iceland, just for funsies, decided they were going to do a genome on their entire country. Now, it wasn't for funsies, obviously. They did it because they have a founder effect, and they want to make sure that they can properly educate people about the risk of essentially marrying your cousin or a second cousin or third cousin. And it cost them only $600 per person. So 2003, $3.8 billion. 2019, $600. Next slide. Now, I don't know about you, but my Facebook clearly hears me talking about genomes a lot, and it decided to advertise to me getting my whole genome sequenced for $300. And so this price just keeps on going way, 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 way down, and we are not even close to where we used to. And so when you think about that, next slide, this follows what we would call a logarithmic curve. And so this is why things have changed dramatically in the span of a very short amount of time. Now, this is a really important graph, and I hate graphs in general when talking to the general population, but this one's a really important one and highlights so much about what I want to talk about. Now you'll notice that y-axis, the vertical line on the left, that each one of those ticks goes up by 10 times. This is not going up by 1, 2, 3, 4, 5. It is 1,000, 10,000, 100,000, million, 10 million, 100 million. So we are going up really, really quickly in the small number of those of that room there. Moore's law, that little dashed line there, this is like for a fun little trivia fact. It was an observation that was made in the 1960s that the power of computers doubles every 2 years, meaning that the cost of computing goes down by half every 2 years. And so computers are getting smarter and smarter and smarter and cheaper and cheaper and cheaper. Now, initially, what limited us in this in terms of cost were our computers. And as we're all aware, our computers are worlds faster and worlds cheaper than they ever used to be. And initially, it kind of followed this curve. And then starting right around 2008, right around when I was finishing med school, the cost plummeted. And it went from, again, only the super rich can do this, to now we get where, again, I'm getting advertised in my Facebook for a few $100. I can get it done for fun whenever I want. And now, as you can see, they stopped this slide here. This is from the NIH. This is a government-funded graph here. They stop it at 1,000. As I had mentioned, we are significantly below that now. Next. And so our knowledge of epilepsy has dramatically changed during this time. Beforehand, we needed to know exactly what we were looking for, and we had to find it in a very meticulous and very, very painful manner. I won't go into it other than to say there's a reason why we only discovered about 10 genes in the first 15 years that we were doing this. Then, right around the time that cost plummeted, we just decided, you know what? We are not going to be really careful about this. We're not going to be really focused. We are going to shotgun, and we're going to look at everything. And we are going to start just basically saying, where's the signal? Where are we seeing consistent differences? And lo and behold, very, very quickly, we found them. And we are now, this slide, this was published in 2016. As I had mentioned, the current number of genes we have on there is 300. So that curve just continues to go up and up and up. Next. And so, as I had mentioned, when I first started my clinic 6 years ago, this was a giant pain, very expensive. Insurance often said no. There were a lot of surprise bills, so a lot of doctors wouldn't even bother ordering it. Took forever, needed to get a blood draw, not fun. Now, next slide. As I had mentioned, I guess I forgot that I did include the name of that gene. That's okay. Every doctor knows about that one. As I had mentioned, worlds easier, to the point that we're now at the point where doctors often, we have this one saying where if you're thinking about it, you just might as well do it, just because it's so easy and it really is almost consequence-free to do it. And certainly it is almost cost-free as well. Next slide. And so we are not on this well-founded body of knowledge that we know everything and we're just kind of connecting the dots. What we're doing is we're actively discovering, and we're working together to discover. And our patients are an important part of that discovery. Every time I discover a patient with a new mutation, well, sometimes it fits in very, very well with what we know about. Sometimes it doesn't. And I have quite a few patients where they are 1 in 10 in the entire world that have anything like this. And we're learning together, quite frankly, because I can't share that information about my patient willy-nilly with the other 9 families. My patients can, and they do. And they kind of work together where we're all discovering this at the same time. Now, the important piece, though, is that we need good data. We need to know that we're all on the same page here, which right now it's a little bit of a wild west because we have a lot of people, and I include doctors in this conversation. This is not just a patient phenomenon. We have a lot of people that think they know and think they have a complete understanding and make what they consider to be definitive diagnoses when it's not that simple. And so that's one important piece. The other important piece is that before, when we were discovering these genes, when it was expensive, we weren't really doing sequencing in people who had relatively mild epilepsy. We were only sequencing people who were struggling, who we didn't have a good answer in. And so whenever we kind of say, okay, people have a mutation in this gene, what do they look like? Well, we're comparing them to this old information, which for the most part was people who are not doing well. Now that we're sequencing people who are doing well, who are not struggling because we can do it more easily, we don't understand what the mild end of the spectrum is for these illnesses. We understand the severe end much, much better than we understand the mild end. Just because, again, we don't, scientists don't dedicate their lives to curing epilepsy that goes away on its own. Scientists dedicate the work, dedicate the money, put all the effort into these people who need help. And now we're testing people. We're getting this information. We're discovering genes that don't necessarily have a severe end of the spectrum, that don't always have a severe outcome. And so I have a lot of patients in my clinic who come to me saying, I got this horrible diagnosis. This gene is associated with epilepsy that causes you to die early. And I'm horrified what this means. And my daughter's doing really well right now. And I tell them, well, your daughter's doing really well. Perhaps this is, maybe your fate isn't as written in your genes as much as you think it is. And again, going back to the patient, going back to the kid, going back to the person with epilepsy, and just saying, if it doesn't fit the gene, believe the person, believe the kid, often more so than we believe the gene. Let's go on to the next slide. Okay, so now that I've done those caveats about it, let's talk about the nuts and bolts of practice. Should I go ahead and get it? And I imagine for most of you, if you're attending on your Wednesday evening, choosing to listen to me, I'm guessing you're already more interested in this than most, and probably the answer for you is probably yes. But let's talk about it. Next slide. So the rule of thumb, if I wanted to give you a simple answer of who should be tested, well, anyone who fits the description potentially for any of the thousands of known epilepsy syndromes or epilepsy presentations that are associated with our epilepsy genes. Now that is the simple answer. Now, in order for that answer to mean anything, you have to know all of the thousands of presentations, which I just had mentioned, changes every single day. And so that's a really hard way to really conceptualize that. So instead, the way that I like to think of it, next slide, is the other way around. Who shouldn't get tested is probably the easier way to think of this. And I will say all of these actually should have the caveat with a few exceptions. Now, a different cause has been proven. Now, I like to tell this story just to highlight how humbling the brain can be. I had an individual who, at the time I had met them, had two reasons for their epilepsy. And made clear, no doubt about it, slam dunk reasons for their epilepsy. And so I'm like, I don't need genetic testing in you. And because I was so focused on those causes, I was focused on treating them. I did not do the family tree on that first visit. And later on, they had mentioned that they had a family member with epilepsy. I'm like, oh, tell me more about this. And sure enough, this particular person had a third reason for their epilepsy because I did their genetic testing, and that was completely unrelated to the other two. So point is, as I had mentioned, the rule of thumb for thinking about it, you might as well just do it as long as you have a team ready to interpret it. For the most part, if you have an abnormal MRI, your genetic testing doesn't add a whole lot. And including in that one patient I had just mentioned, they had an abnormal MRI. That was one of the reasons I wasn't interested in doing genetic testing. For the most part, the MRI really tells me 99% of what I need to know. And in very few cases does at least add anything, other than sometimes it completes the story for us. In well-controlled epilepsy, I share this primarily because, as I had mentioned, most of the time you get bad news when it comes to genetic testing. They're going to tell you this is associated with this kind of bad epilepsy and this kind of bad epilepsy. They're not going to tell you that, oh yeah, but half of them actually outgrow their epilepsy and everything's fine, because those people weren't tested. And so what I find is that when we get some of those genes where now I'm convinced that this gene now has a milder end of the presentation, I just tend to just say, you know what, if you're doing well with your epilepsy, you probably are not going to stand to gain anything by doing your genetic testing, except for scaring yourself. Febrile seizures only. Now, I think that by the time I retire, which hopefully is many years away, by the time I retire, genetic testing will be done for febrile seizures only. It is not ready for that right now, simply because there are a lot of people who have just febrile seizures. And by that, I mean having seizures with fever between the ages of 6 months to 5 years, who have just during that time and no other time in their lives, and then outgrow it never have any problems for the rest of their life. There are people who have the exact same mutation, the exact same change in the exact same gene as people who have horrible epilepsy. And our genetic testing is not currently equipped to distinguish between those people. And so again, febrile seizures only is like the king, in my view, of you're only going to get bad news, and that bad news isn't necessarily bad. It's just that it sounds bad, but it doesn't mean what we think it means. And so I've been trying to caution doctors to be very careful about doing it in that test. And then there are certain epilepsy syndromes. These are the more common ones, actually, that actually used to be called the genetic generalized epilepsies, GGEs, that we all assumed are genetic because they run in families. But lo and behold, we have been doing the DNA sequencing in these, and scientists have not figured them out. We're not finding basically any genes in these people. And so for specific epilepsy syndromes, again, you can read them there. If you have one of those or your loved one has one of those, you'll know what I'm talking about. I don't want to teach about those particular epilepsy syndromes. Point is, for those specifically, we're not actually finding any genes, which is much to common sense's surprise, to be honest. Next slide. The rule of thumb for me, and kind of the ones that I really kind of push for it, is unexplained refractory epilepsy. If we're struggling with your seizures and I have no clear good reason for it, then we should be doing it. And my definition of struggling gets a little bit milder every time. Sometimes it's just the Keppra, I thought this was going to be completely smooth sailing, but I'm needing surprisingly high doses of your first medication. And that is enough for me to cross my threshold for testing. Sometimes it's just on medication number 2. But for the most part, for me, if I'm on medication number 3, that is one of the things in my mental checklist about refractory epilepsy, along with the question of epilepsy surgery an option, which whole other conversation. Epilepsy plus. So essentially any other weird genetic thing, horseshoe kidney, a heart valve abnormality, birthmarks, and I mean by birthmarks, I mean not the common ones, but atypical ones. Just anything else that is unusual and something present from birth. Intellectual disability is actually the single biggest one. We've actually gotten to the point that intellectual disability plus epilepsy is one of the highest yields. And by yield, I mean the most likely to find an answer with doing genetic testing. And also the younger you are when you first had seizures. And we're actually currently estimating that the rate of finding an explanation from genetic testing in seizures under the age of 2 is probably about 2/3. Very high likelihood of finding something. Certain epilepsy syndromes, I just list them there. If you don't know what they are, again, they probably don't afflict you or a family member, but those are the hallmark ones where we do want to get genetic testing because it often does make a difference. Seizures run in the family. It's one of the more common ones that I get consulted for. As I just highlighted, and some of the more common epilepsy syndromes that run in the family, we don't find anything, but it's worth at least taking a look there because that's often where we discover, as I mentioned, I learn about epilepsy every time we find another family with one of these genes. And it also helps with our family planning. I think that, I would say that before I had a kid, I would have said that if I had mild epilepsy as a kid and now I'm having a kid, it doesn't matter. If my kid has mild epilepsy, everything will be fine. Now I have a kid and now it's a different story where I'm like, no, I kind of want to know, I need to brace myself. And it's something where it's reasonable to say, let's test you because you have epilepsy, and then we can test your kid. And if your kid doesn't have the same mutation as you, I can say you don't got to worry about this, which again, as a parent, I appreciate that. Anyway, next slide. The downside, again, I really already talked about it. As long as your team is prepared to interpret the results intelligently, and that usually means a geneticist. I'm very selfish in saying that, I often need to be involved in it just because I've seen some really smart people make some false conclusions. But there are multiple genetic neurologists out there who are well poised and well prepared to answer that question. And I work with multiple geneticists in town. Most of them have my email address, and I can work with them if there are any questions. As I had mentioned, I mean, other than that, the cost is the only risk. At the moment, this is a common question I get. Could this impact my ability to get insurance in the future? The answer is, according to the American Affordable Care Act, the answer is no. However, that is just as we are all aware that could change at any time, and laws could be made that could be an impact. But at the moment, the answer is no. It cannot impact insurability issues. So it shouldn't be anything other than just, again, making sure you're someone prepared to answer. Next slide. So let's now just talk about what we can do with the information. Next slide. So in some cases, it gives us an idea. It lets us predict that perhaps the future might get some challenges that might be coming. So like for SCN1A, we have a general idea of the time frame where seizures are the hardest. In some cases, actually I have now multiple genes. I should have added some beyond SLC2A1, where we know that the other things, movement disorders are abnormal movements that aren't seizures. There's a very short version of them, where I can counsel them when they have that abnormal test to say, hey, you might see these certain types of movements. And if you do, don't assume they're seizures. They might be involuntary, but they're not seizure. And that has been tremendously helpful in the few people that have had those particular genes. It often means we have to look for other problems. Like with GRIN2A, we know we need to do overnight EEG. And the other way around, it might also predict that things will get better. There are some of them where we know that the seizures are worst in certain time frames. And so for the ones listed there, we know that the infancy period is typically the hardest period, and then seizures get better as they get older. For other ones, we know that early childhood is typically the hardest one. For others, we know that they'll go long stretches in between where everything is fine. And so maybe we should, and then they'll have hard to control clusters. So maybe we focus on treating the clusters and try to avoid having too many medications in between those clusters. And they kind of help tailor our treatment a little bit more custom. Next slide. We are getting better and better at predicting which medications are best for these. And the parents are head and shoulders ahead of the scientists at figuring this out. The story I love to tell about this is that I had a patient with a KCNQ2 mutation, which you see on that list there, when I was in residency, and there was a brand new medication that targeted the KCNQ2 receptor. And I was so excited to prescribe it because that was kind of like, it's a lock and key to me, right? You have a problem with this receptor, this treatment treats that receptor, bam, you're going to be fixed. I'm really excited about this. And the mom tells me, she's like, I'm on Facebook with these other moms with KCNQ2 disorders, and they're telling me that Trileptal is working really, really well for them. And I said, well, fine, that's a safe, easy medication. It's cheap. I can get insurance to approve it. Let's give it a shot. And sure enough, it worked like gangbusters. And it took about another 5 years before that was actually published in a scientific journal. It was figured out on Facebook 5 years before the scientists had figured it out. And thankfully, my family found it so that that patient benefited directly from that. And now for every KCNQ2, I'm so excited to run to that, and I've seen lots of good responses there. And then there are others where we know there are certain medications that make it worse. And I'm going to keep on moving because I just noticed I'm a little bit behind on my time because I get wrapped up in this stuff. Next slide. We are now getting to the point that there are some medications that are custom tailored specific, only ever used with certain mutations. And this list will grow, I'm hoping, with every year to come. Again, I don't want to run through the list here, just to know that we're getting there. There are 300 genes I had mentioned. This is what, maybe 15 that it actually makes a difference. I'm optimistic that this will only continue to grow. Next slide. But by far the most consistent benefit, where everyone really appreciates it, and that story I told about KCNQ2 really highlights it. Whenever you get an answer, what you get to do is you find a community. You find a community of other people that are dealing with the same struggles, dealing with the same challenges, who are really engaged and dying to share their knowledge because they've gone through heck often with their own kid or their own family member or themselves. And they are really hoping that the lessons that they learn could be applied to other people. And I learn from them just as much as the patients do. And without fail, this is what people really appreciate, often more than any other medical benefit I can offer. Next slide. And so let's take a quick glimpse into the future, because I'm going to be horribly wrong. Let me take a stab at it. Next slide. So we already know that the better, cheaper testing, that this is only, again, it's only going to get cheaper. We know that. And it's already at the point that it's cheaper than getting an MRI. And so we're going to get to the point where it's going to be a reflex thing. Instead of getting an MRI, we might even do the genome first, and you get the results back in 24 hours. I'm betting that's going to happen in my time before I retire. We're going to be doing it earlier and earlier and earlier. And again, perhaps even at birth at some point. I don't think we're anywhere. I don't think that'll be in my lifetime. I hope not, actually. But I do know in the case of diseases where we don't know what the future looks like, we're going to be doing testing much, much more early. I guarantee you we're going to be having some more custom tailored medications, and it's definitely having some more non-seizure medications, more custom treatment options. But what gets people most excited is gene therapy. And, we only have a tiny amount of time for that. And I'll just say that I thought we were over a decade away when I started this, my clinic about 6 years ago. And I have been approached starting about a year ago about the first opportunities in gene therapy and epilepsy. And so we are ahead of where I thought we would be. Now, it's not universal. There are over 300 genes. I'm aware about 5 that there is any kind of gene therapies imminent. But we're getting to the point that we are starting to use genetic tools to treat neurological disease. It's not yet there for epilepsy, but it is there for other neurological diseases. And it's only a matter of time before epilepsy is going to be there as well. And I can't wait. I am very, very excited to give it a try. I think it's going to be worlds more complicated than what's already been done, for many reasons that I hope one day I'll get a forum where I can just wax philosophical without slides and talk about where I see the challenges being. But once we figure it out, this is going to be a whole new ball game about how we're treating epilepsy, and I'm really excited about it. And I think I actually then finished on time. I wanted 15 minutes for questions, so I think we're good.
Thank you so much for everything that you've offered us tonight. It's been amazing. You are such an expert, so I can't thank you enough. We are going to go here to our Q&A. It looks like my daughter has a 5S5 gene mutation, was correlated with this gene mutation. We were told autism could be, but she's also recently been diagnosed with a tic-tac disorder. She's still having seizures. I cannot stop. I had epilepsy and I did not grow out of it. Why is her so different than mine? So maybe going back to the difference between a mother's epilepsy between her daughters.
Yeah, so I will start by saying DS5 is not one that I know off the top of my head.
Okay.
So it's not a common one. I should know. My initials are DS, right? So I feel like I should be more knowledgeable on that one. I don't know that one off the top of my head. Now, I also want to note that the question specifically states mutation. Now, mutation doesn't say whether or not it is a problem or whether it's a difference. Mutation just means it looks different. I'm going to assume that in this case that it is what we call a pathological mutation. I would need to first double check. There is a website called omim.org, omim.org. And I would enter DS5 on there. And actually, I mean, unless we have other, I will do this live just to kind of show what I do. I'm going to omim.org, as I just mentioned, typing in DS5. And actually, there's no entries for DS5. And so there must have been a typo in that gene because there is no gene DS5. So I would need to know, again, it really boils down to the specifics. As you had highlighted, DEP-DC5. Okay, that I know quite a bit about. DEP-DC5 is the most common cause of focal epilepsy in families. It is the best metaphor. I don't have a good metaphor for it, actually. There is a disease entity, as Nikki had mentioned, I run a TSC clinic. It's closely related to TSC in that DEPDC5 is a gene that controls the brakes in cell growth. And you need two of them to tell the cells not to grow. For most of your cells, that's fine. But once you get that second hit, now you have no brakes. And that cell then starts to grow. For DEPDC5, that usually means you get something called a focal cortical dysplasia. And focal cortical dysplasia is going to be a something that you're born with, happens early in birth, that the cells don't line up right, and really the only thing they do is seize. Now, depending on how many of those second hits you get and where that focal cortical dysplasia is, it can determine how severe the seizures are, what the seizures look like, and what other neurological problems you have. And so that one is known to be very, very variable from mother to daughter and parent to child and across the board. That one is one where you only need one bad copy. I see the question about CHD2. The best resource, I don't have actually, I don't know off the top of my head the parental resource. I go to a website called epilepsygenetics.net. And CHD2, if I remember correctly, does have something what they call an epilepsiome. This was the guy I trained under, Dr. Helbig. His dream was to have a well-curated library for all the most common ones, and then there were just too many genes, and he gave up on it because it was too much work. But it's a wonderful starting point for kind of just getting what are people generally saying about CHD2. My experience with CHD2 is that the seizures are not the hardest to control, but what we call the epileptic encephalopathy is. And by what that means is that the development, the cognition, often is a little bit more of a struggle, but it seems to respond in my experience with my few patients that that they've done better with seizure medications, more so than their seizures themselves. CHD2 can also look like Dravet syndrome, some other epilepsy types where I have not personally seen it, but I know it can look like. Middle-aged female with temporal lobe epilepsy since age 15, well-controlled on that was already, there it is. No one else in the family has epilepsy. Does it make sense to undergo genetic testing? In this case, I would say probably is not going to add anything. If things are going well, I think the one big question we always talk about with temporal lobe epilepsy is epilepsy surgery an option? The genetic testing wouldn't really weigh into that. If your seizures are already doing well, Oxtellar has no known long-term side effects. It's something that a woman can safely get pregnant on without any issues. I would say that the genetic testing probably wouldn't necessarily impact things. At the age of onset of 15, it's less likely. It's actually only less than a 5% chance we're going to find something when you're that old with your first seizure. And so it's low yield as well. Now, that said, less than 5% isn't 0%. And so it might be worth pursuing, although I would say that's not necessarily the one that I would necessarily recommend. ALG6, don't know off the top of my head. A known gene connected to CDG. I'm guessing by CDG mean congenital disorders of glycosylation. ALG6 must be, I don't know all the genes off the top of my head for the congenital disorders of glycosylation. They are, as a family, the rarest collection of the inborn errors of metabolism. I have seen 3 in my career, so they're very, very rare. And in terms of what's known about it, again, I was hoping to do a live demonstration by going through OMIM and telling you what I'm seeing and corresponding it. But omim.org is a great place to start. It's very scientific, unfortunately, but a great place to start to kind of get an idea what the scientific community is saying about it. And this is all public, by the way. Like, none of this do you need to pay for anything. This is all government funded. My son's epilepsy is idiopathic and his seizures are fairly controlled with medication, but he does have breakthrough seizures every couple of years. Because we're constantly anticipating the next seizure, we want answers. Is genetic testing the way to go? Now, I would need to know a few more bits of information. The short answer is yes. I think that genetic testing could be helpful here. It may not be enough to get you across the finish line to seizure freedom. In terms of what I'd want to know is the age of onset. Was that seizure under the age of 2? Under the age of 2, as I had mentioned, very high likelihood of finding something. Use the word idiopathic, so I'm assuming his MRI is normal. And so, yeah, he is the type of person that I typically would be interested in genetic testing. The other piece about breakthrough seizures every couple of years, how much of that is related to triggers? A lot of times, again, the milder epilepsies have a harder time finding finding genes associated with them. And particularly if they're related to, he got the flu, and if he gets a serious illness, every couple of years we see something, a little bit lower likelihood. Oh, great question. Is focal cortical dysplasia caused by a genetic mutation? Or are there other explanations? That is a topic for an entire lecture on itself. There are scientists who believe that all focal cortical dysplasias are due to genetic mutations, but they may not be germline. Now, what do I mean by that? Not the type of mutations you can pass on to your kids. There are people that have just taken all the tissue from focal cortical dysplasias that were removed and sequenced every single one of their cells and have found the mutations and the genes associated with it in like one of those thousands and thousands of cells. And so, again, that's a short version of a very interesting topic where the short answer to your question is there are other explanations, but doctors are very interested in seeing, is this all genetic? We had genetic testing through Children's Hospital about 5 years ago. My daughter has Pediatric syndrome. She had 17 seizures in 5 years, but has been seizure-free 5 years now, except for occasional brief absence seizures around her menses. Her grandfather had some form of childhood epilepsy and is bipolar, and they have profound behavioral issues. Is it worth retesting? Now, for Paniatopoulos, there's not any genes that I'm aware of necessarily that immediately come to mind as connected. So it's not like she is an obvious candidate where I would say she absolutely needs it. That said, 5 years, there have been a significant number of genes that have been discovered. As I mentioned, 6 years ago, the panel was about 60 genes, and now we're at 300. She may not be necessarily the clearest candidate for it, but I would say there's probably some utility there, particularly because you're still having some struggles and because there does seem to be a bit of a family history there. Oh, I'm so sad. Our daughter passed away from this, from the congenital disorder of glycosylation at 21. Yes. Yeah, it is. It's a very challenging disease, and it's been hard for me in taking care of those, and my heart goes out to you. And I share this. My best friend, he had a sibling who passed away from another rare neurological disorder, and there now is gene therapy for that specific disorder. And I remember telling his mother about this, and this happened 40 years ago, and it's still breaks her heart. And I thought it would bring joy and solace, and I hope that what you went through brings attention to what was going on, so that the next person doesn't have to pass away from it. How do I make the determination to use panel testing versus whole exome? I personally don't order whole exomes. I utilize our genetic specialists just because there are a number of other challenges that go into it, and they have the resources to follow up on those results and do those repeat analyses over the years. I only do panels. And in people where it just smells of epilepsy, it smells like genetics, and that first panel comes up negative, I will move on very quickly to refer them for their whole exome sequencing. When my daughter was first diagnosed with DEPT-D5, it was considered rare. Is it more common now? Fantastic question. And I could also spend a lot of time on that one. It wasn't tested very much. It wasn't on most of those panels, even though when you looked in the science, it was the most common cause of genetic focal epilepsy. We had to specifically request it. And so the incidence is probably much, much higher than it was. And it certainly is more, I would say it is more recognized now because it's on more of our panels. What medicine would you suggest use that wouldn't raise ammonia levels as you do with many, as many do with urea cycle disorders? I'm guessing you're talking specifically about epilepsy medications. I am a big fan of obviously of Keppra, because Keppra, as most of you will know, is low. I'm sure that's already been tried. Onfi is another one that I don't really see, or clobazam doesn't really raise ammonia levels. The main one, obviously, that we avoid is Depakote, and that's followed probably closely, or valproic acid. We also will avoid phenobarbital, or we would also likely avoid felbamate or felbutol. And topiramate, it's not talked about a whole lot, but topiramate or Topamax actually has a meaningful incidence of elevated ammonia. Thank you for sharing more of your. I'm not going to say your name, but I'm reading all of it, and I appreciate sharing your story. My daughter has Aicardi syndrome and refractory epilepsy. We have had genetic testing, and we're told it would be retested every 2 years. Who reviews them? Her main doctor, the geneticist. Aicardi syndrome is a fascinating genetic disorder. Aicardi is only present in girls, so we know where it's coming from, right? It has to be on that X chromosome, and yet we still haven't found it. And it's relatively common. And so it's one that I'm still shocked we haven't quite found. In your specific case, we're going to have a breakthrough one of these days, and we're going to figure it out. Aicardi is so distinguishable, so clear compared to so many of these other syndromes. There has to be a simple explanation for it genetically, and we're going to find it one of these days. And I assume if you're connected with the Aicardi community, you're going to hear about it. In terms of, that's not what your question was. How does that automatic review occur? The answer is whoever is doing their testing does an ongoing review continuously, actually. And anytime that variant comes up, they're going to wind up, or anything that changes the way the data is going to be interpreted, they output an updated, they output an updated report that goes to whoever orders it. So it's actually taking place at the level of the genetic testing company. Now, that said, I don't always assume that they're doing it. Yes, they say, and I often get those reports. But anytime I get a patient where, again, it still smells like it, I still think this is what's going on, I do intermittently check in and just say, hey, just checking in on whomever, do we have any updated response? And for the most part, especially if you order regularly from them, it's not too hard to get a response from them. And so your answer to more completely answer your question, it's a combination, should be happening automatically at the gene testing company level, but your doctor can also help out there.
I really appreciate, oh my gosh. Dr. Smith, you've been amazing tonight. You've been so helpful, and you've given all of us a great insight into all the different aspects of how genetics can play into this. So I can't thank you enough. Other than that, if you have any genetic issues or any questions you have on client services, we're very happy to help. So we're going to have another one in November with one of Dr. Smith's colleagues, Dr. Ann Hempel, on the impact of epilepsy on cognition and memory. So we hope you can all join us there. Thank you so much, Dr. Smith.
Thanks everyone for coming and for spending your Wednesday evening in front of a computer with me.